are the nuclei. Marked cells are on the bottom left. There's a good bit of detail in this chapter. There's also a chart provided in the files on Canvas that will help you order material should you choose to use it. Our vision is extended by microscopes and telescopes, our senses of smell and taste by machines that measure chemicals. Remember, science is an accumulation of knowledge. Our understanding of the world around us alters as our information base alters. Could we have known cells existed before there was a way to view very tiny things? Could the cell theory have been developed? After the microscope was invented, Robert Hooke discovered cells in 1665. It took many more years and many scientists making observations before the cell theory was developed, which in part states all living things are composed of cells and all cells come from other cells. That was in the mid-1800s, 200 years after the discovery of cells. It took 200 years to gather enough information to come up with the cell theory. Think back to the last section. What type of science was being used by these scientists? Yes, discovery science. Inductive reasoning was used, forming general principles from a large number of observations. The original microscopes used mirrors to reflect sunlight through the specimen. Today, we use electricity, although you can still find some scopes meant for children or for field work that use mirrors. Compound light microscopes allow you to watch living organisms move. 1950s. That's less than 70 years ago. We've made lots of discoveries using electron microscopes already. Where will we be 200 years from its discovery? How do you think a paramecium in pond water will behave with a stream of electrons passing through it instead of light? That's right, it'll have to be dead under this scope. Scanning electron microscope is useful only for surfaces of cells and other structures. An electron stream hits the surface and the pattern forms the image. Electromagnets magnify and focus the image, which appears three-dimensional. Transmission electron microscope is used to study internal structures of cells. Extremely thin sections of the specimen are cut and stained with heavy metals, like gold. The electron stream is sent through the specimen rather than over its surface. Electromagnets magnify and focus the image. Note that magnification is how big you can make it, while resolution is how sharp the image is. Euglena under all three microscopes, light microscope, scanning electron microscope, and transmission electron microscope. You can see that you can get different information from each. You do not need to know these numbers, but do note that they overlap. You can see a frog egg with the naked eye, but you can see more details under a light microscope. Also note that with an electron microscope, you can see viruses and even small molecules. I expect that within your lifespan, someone, maybe you, will invent a microscope that can see individual atoms. Which microscope is the one which provided all the current images of COVID-19? Scanning electron microscope, showing a three-dimensional surface view. Can you think of any of those necessary components? DNA? Protein molecules? Organelles? Those wastes and that nutrition must come through the cell membrane to go in and out. What's a surface to volume ratio? How much surface compared to how much volume? Think of the room you're in. Now visualize it full of people, really full, packed like sardines. All the needs of the people must go through that one door. Will the people furthest from the door ever get food? Will their waste get to the door and out, or is too much going on for the access? Cells are similar, although food and waste can travel directly through the membrane. We could do that to your room, but how many holes can we have open without damaging the integrity of the structure? The same holds true of the cell. Only so much can go through at any given time. If the cell gets too big, it will not be able to take care of itself. Compare the ratios. On the single large cell, there are 2 parts of surface for each part of volume. For the multiple smaller cells with the same total volume, there are 6 parts surface to 1 part volume. Remember, surface is calculated by height times width times the number of sides. Volume is height times width times length. The plasma or cell membrane isn't just a divider. It's like a wall. Membranes are places. Lots of processes happen on or across membranes. While the phospholipid bilayer we saw in the last chapter is the base for the membrane, there are lots of other molecules embedded in it. Some protrude on one side or the other, while some go all the way through. These contribute to the functioning of the cell or organelle. There are several different proteins shown in this membrane. A channel protein is like an open door through the membrane. Anything that fits can come in or go out. Other proteins may be embedded enzymes. Still more might act as restricted channels, only allowing certain molecules through. Some may transmit signals from one side to the other. There's a lot of variety. We briefly mentioned these cell types in the overview given us in Chapter 1. Now it's time to get more in depth, a lot more. This chapter will focus on eukaryotic cells, though. Common features between the 2 cell types: DNA, plasma or cell membrane, ribosomes, and cytoplasm. Differences: eukaryotic has a nucleus and membrane-bound organelles, including the nucleus, while prokaryotes don't. Prokaryotic domains are archaea and bacteria, and eukaryotic domain is eukarya. Prokaryotic cells are smaller and simpler than eukaryotic cells. Knowing that will help you remember why, because eukaryotes have more stuff, nucleus and organelles, that prokaryotes don't. The plasma membrane simply contains the cell, much like your skin contains you. DNA for all cells contain genes, which code for proteins. And all cells need ribosomes, which are tiny structures that use the instructions from the genes to make the proteins. These are organelles, but they are not membrane-bound organelles. This is why we must be specific when saying what they do or don't have. You can't just say prokaryotes don't have organelles. They do. A diagram on the left and an electron micrograph on the right show a typical prokaryotic cell. While ribosomes are necessary, some structures are optional by species. Fimbriae may help a bacterium to attach. A capsule may provide protection. There may be one or many flagella. The nucleoid region is where the DNA is, but there's no membrane around it, so it's not the same as a nucleus. Add all these possibilities to your compare and contrast of prokaryotic and eukaryotic cells. There are lots of parts to a eukaryotic cell, and we're going to need to know the major ones. The compartmentalized organelles are the membrane-bound organelles. The membrane separates them into individual compartments. On your compare and contrast, you won't need to list them. Saying membrane-bound organelles covers them all, except for the nucleus. Compare the animal cell on the top to the plant cell on the bottom. They contain some organelles the same and some different. Each organelle is like a little organ. Just as each of your internal organs has a job, each organelle has a job. You may wish to refer back to these pictures as we covered the major organelles. This is an animal cell. This is a plant cell. Since the nucleus has the DNA in it, it's the organelle responsible for controlling the cell's activities. It directs protein synthesis. What does synthesis mean? To make. The nucleus does not have all the DNA in a cell. 2 organelles are directed by their own DNA. These are the mitochondria and the chloroplast. For most of the cell's life, the DNA is in chromatin form, but that will change when it's time for the cell to divide. That's when the chromosomes condense into the structures you can see under the light microscope. The nuclear envelope is double-walled with pores. What are the pores for? To let things go in and out. What things? Either completed proteins or at least the directions to make them outside. The endomembrane system we'll go more in depth on shortly. The nucleolus produces ribosomal building blocks. It's an inclusion in the nucleus. rRNA is ribosomal RNA. There will be 3 types of RNA we'll discuss this semester. Transmission electron micrograph left and diagram right of the nucleus. Note that both the nuclear envelope and the adjoining rough endoplasmic reticulum have ribosomes attached to them. These are called bound ribosomes, as they are bound to the membrane. As discussed earlier, they may be organelles, but they aren't membrane-bound organelles. rRNA is ribosomal RNA. This makes up part of the ribosome itself. Ribosomes may be free-floating in the cytoplasm or bound, attached to an organelle, such as to the nuclear envelope. The ribosome is composed of 2 subunits, one of which is larger than the other. They come together to do protein synthesis. The transmission electron micrograph is a cross-section of folded, rough endoplasmic reticulum showing both bound and free ribosomes. A group of organelles working together toward a common function. Some are physically attached to each other. Others are only connected by function. All are working together. Together, they form the endomembrane system. What's the common function? Synthesis, storage, and export of molecules, proteins. The organelles in purple are linked physically. All of them on this list are linked by function. These are the organelles of the endomembrane system. Endo means inside. There are 2 types of endoplasmic reticulum, rough ER and smooth ER. They are so named based on whether or not they carry bound ribosomes. Rough does, smooth does not. You can see in this drawing how the ER is a continuation of the nuclear envelope, part of the endomembrane system. Smooth ER is the tubular one, smooth because no ribosomes. Rough ER, which is what we'll look at next, is more envelope-like in structure, being in folds. You can see in this drawing how the ER is a continuation of the nuclear envelope, part of the endomembrane system. Smooth ER is the tubular one, smooth because no ribosomes. Rough ER is more envelope-like in structure, being in folds. This organelle has more than one function. Number 1, the main function is to synthesize lipids. What does synthesize mean again? To make. So yes, it makes lipids. Each type of cell, depending on where it is in the body, will make a different type of lipid. What are some types of lipids? Steroids, hormones, fats, phospholipids, waxes. Also, this organelle has more than one function. Number 1, the main function is to synthesize lipids. What does synthesize mean again? To make. So yes, it makes lipids. Each type of cell, depending on where it is in the body, will make a different type of lipid. What are some types of lipids? Steroids, hormones, fats, phospholipids, waxes. Number 2, also, cells in the liver have smooth ER that detoxifies, removes drugs. Cells that do this and are exposed to large amounts of toxins respond by developing more ER for the job. The side effect of this is that the organism becomes tolerant of the drug or alcohol or toxin and also of related compounds. But remember, the cell can only get so big, surface to volume ratio, so it will reach a point where it can't add more smooth ER to take care of a drug or drinking problem. Tolerance is a warning sign. Liver disease can follow. Number 3, in muscle cells, smooth ER stores calcium ions. These are needed to trigger contractions of the muscles. Notice that the amount of ER and the primary function of ER may change with the type of cell. And all of these functions are facilitated by the embedded enzymes. If you're filling out the chart, note that the structure is smooth, interconnecting membrane tubules, and there are 3 functions. Why do you think the cell might need more membrane? To send the proteins elsewhere in the cell, membrane bubbles called transport vesicles bud off the rough ER. If that membrane wasn't replenished, the rough ER would quickly be used up. What stages does the primary protein need to go through to make it a functional protein? Review Chapter 3 for that. What does a ribosome do? Protein synthesis. Uses the instructions from the genes to make the protein's primary structure. Note the ribosome on the rough ER synthesizing a polypeptide, the primary structure of a protein. This polypeptide completes the subsequent stages inside the rough ER. What stages are these? Secondary, tertiary, quaternary? What happens in each? Review Chapter 3. Transport vesicles bud off the main rough ER and transports proteins elsewhere in the cell. So why do you think the rough ER needs to make more membrane? The secretory protein illustrated here is an example of a protein created inside a cell that is sent outside the cell to work, such as insulin. Note the short chains of sugars added to the protein in the example. Adding sugars to a protein makes it a glycoprotein. The pinched off section of membrane with the glycoprotein in it is now a transport vesicle. Where is it going? To the Golgi apparatus. While the organelles we've looked at so far have interconnecting membranes, the Golgi is more like a stack of flat discs, like a stack of pita bread. One side receives the transport vesicles. Proteins may be modified by repackaging with other items for the same destination or by other means. They may move through each layer of the Golgi by transport vesicles, or the entire stack may move with the contents to the sending end of the Golgi. Finished products may be marked to go to other organelles or to the plasma membrane for secretion outside the cell. Cells that do little secretion will have fewer Golgi than cells which have secretion as a major function. The Golgi apparatus as both diagram and micrograph. Hydrolytic enzymes. What are they? What word have you learned that is close to that? Hydrolysis, breaking down of monomers. Lysis, lyso, breakdown. Opposite of dehydration reaction. So these organelles have enzymes that can reduce macromolecules to monomers. If a protist engulfs a food particle, a lysosome can digest it, releasing the nutrients to the cell for use. Bacterial cells engulfed by our white blood cells are destroyed in the same manner by the lysosomes within the white blood cell. Damaged organelles can fuse with the lysosome and be digested, with the molecules being reused within the cell. What do you think might happen if a lysosome were to break open within the cell, releasing these hydrolytic enzymes? Does the nickname suicide capsule give you a hint? The release of enzymes that can digest organelles wouldn't be something a cell could live through. Note that this is a small membrane sac, but so was a transport vesicle, wasn't it? You'll need to add that this is a membrane sac with hydrolytic enzymes to keep them from getting mixed up. Items broken down by the lysosome can be brought to it from two directions. Internally, damaged organelles are recycled. From outside the cell, food particles or threatening bacteria may be brought in for digestion and destruction. Note the food vacuole shown. We'll look at those shortly. So what's the big deal if you're missing one little enzyme? If you're missing one, whatever that enzyme should have been breaking down won't get broken down. Remember that enzymes can't do the job of a different enzyme. In Pompei's disease, it's the animal storage polysaccharide glycogen. The body can store it, but can't come back and break it down for use later. So glycogen accumulates in muscle and liver in amounts that damage the individual cell first, then the tissue and organ. In Tay-Sachs, the enzyme needed normally breaks down a lipid. Not a big problem until you realize it's one in nerve cells. So nerve cells, including brain cells, swell with undigested lipids, causing permanent damage. Most of these diseases are fatal in childhood, or they used to be. We have advanced medically enough that if we can identify the problem, we can provide the child with the needed enzymes. We saw the food vacuole in the last section. Go back. See, it's small. It's larger than. We saw the food vacuole in the last section. Go back and see. It's larger than the transport vesicles, but the structure is similar. To differentiate it, make sure to say it's a large membrane sac. The central vacuole in plants can function as a large lysosome. It can also enlarge with water or store needed chemicals or wastes. Some contain pigments or defensive poisons against plant eaters. The contractile vacuole, such as those in a paramecium, expels excess water absorbed by the water-dwelling organisms. Without it, the cell would swell and burst. Note how much space the central vacuole takes up in this particular cell. It will vary by cell type. The starch in your potato is stored in a multitude of cells' central vacuoles. On the contractile vacuole, the spokes stick out through the cell, absorbing extra water. The vacuole then expels the water from the cell by contracting. View the video link to watch contractile vacuoles at work. Structural: nuclear envelope, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Functional: ER to Golgi to other destinations, vesicles, lysosomes, vacuoles, all connected by function. The nucleus is the brain of the system. It has the directions to make the proteins. Those directions go to the ribosomes on the rough ER. The protein polypeptide is formed and passed into the rough ER. It gets some modifications done. Then it's packaged up into a transport vesicle and sent on its merry way. The rough ER replaces the membrane. Wait, how? Well, the smooth ER was synthesizing lipids, right? Guess what we need for more membrane? Yep, lipids in the form of phospholipids. In the meantime, the protein in the transport vesicle goes to the Golgi, where it gets finished, then packed up and sent where it's needed. If it gets messed up somehow, it gets sent over to the lysosome, the recycling center. Note, we're going to look at some more organelles, but we're done with those in the endomembrane system. You'll need to know which ones are in the system, which aren't, and if it's structural or functional. Here's the sequence as a visual. The nucleus provides directions in the form of mRNA. That goes to bound ribosomes on the rough endoplasmic reticulum. It forms the polypeptide, inserting it into the rough ER. The rough ER then assists it in folding into a working protein, ships it off in a transport vesicle. It replaces the vesicle membrane with lipids from the smooth endoplasmic reticulum. The transport vesicle goes to the Golgi apparatus, entering from side of the stack of flattened membrane pockets. Here's the sequence as a visual. The nucleus provides directions in the form of mRNA. That goes to bound ribosomes on the rough endoplasmic reticulum and forms a polypeptide, inserting into the rough ER. The rough ER then assists it in folding into a working protein and ships it off in a transport vesicle. It replaces the vesicle membrane with lipids from the smooth endoplasmic reticulum. The transport vesicle goes to the Golgi apparatus, entering from one side of the stack of flattened membrane pockets. It gets further modification and is shipped out in another vesicle from the opposite side of the Golgi to its final destination. If it got messed up, though, it gets shipped over to the lysosome to be recycled. These organelles do their own jobs, which have nothing to do with protein processing. ATP, or adenosine triphosphate, is the main source of energy for cellular work. The cell takes the food molecules, breaks them down, and stores the energy as ATP molecules, which can be utilized as energy throughout the cell. Both animals and plants need mitochondria. Okay, look how the mitochondria is structured. It has a double membrane similar to the nucleus. You have an inner membrane space, or a space between the membranes, but the inner membrane is not the same as the outer. It's folded and folded, as if it's much bigger than the outer and had to be folded up to fit inside. Those folds are called cristae. Inside the inner membrane is a fluid called the mitochondrial matrix. So double membranes on the outside and two compartments, the inner membrane space and the matrix. That's the structure. The chemical reactions of cellular respiration occur both in the matrix and on the cristae. Since the membrane itself is a place where a reaction happens, think about why the folds are needed here. Yes, to increase the amount of membrane available for these reactions to take place. A whole chapter is devoted to cellular respiration with lots of details to delight you, but that's chapter 6. We won't discuss all that here. Since it's the organelle of photosynthesis, it's found only in organisms that photosynthesize. This is another organelle with a double membrane, complete with an inner membrane space. The thick fluid inside is the stroma. In the stroma are folded and stacked membranes called thylakoids, with the stacks being called grana. Photosynthesis takes place across the thylakoid membrane, with reactions on it and on both sides, in both the inner membrane space and the stroma. We have a whole chapter on this organelle too, so won't go into depth here. Wait for chapter 7. For those of you who are not taking lab, here's what chloroplasts look like under a microscope, the green balls. These are plant cells. You can tell not only by the chloroplast, but by the arrangement of the cells and the presence of a cell wall. Plant cells look like they were placed by a brick mason, sometimes a slightly drunk brick mason, but still fairly regular in size and placement. Remember that both the mitochondria and chloroplasts had their own DNA? And now we know that they both have double membranes as well. There's a theory that accounts for these features. Remember the difference between a scientific theory, a hypothesis, and a lay theory? This is a scientific theory. The endosymbiotic theory states that these two organelles were once each free-living, single-celled prokaryotic organisms. The ancestor of the mitochondria was an oxygen-using energy converter, and the ancestor of the chloroplast was an oxygen-producing photosynthesizer. The ancestors were enclosed in another cell, likely an archaean, either initially consumed as a potential food source or they were invading the cell. but stayed when they were found to be useful. Support for this theory includes both organelles have their own DNA, both organelles have their own ribosomes, and these ribosomes are more like prokaryotic type than eukaryotic. Both of these organelles manage their own reproduction, which they do like a prokaryotic cell rather than a eukaryotic cell. The word endosymbiotic can be broken down. The prefix endo means inside. Symbiosis is when two or more species have a long interrelationship with each other, often to the point of not being able to survive without each other. An example of this is termites. They eat wood, but they can't digest it. They have a tiny protist living inside their gut that digests the wood for them. The protist gets a nice place to live with food delivered, and the termites get to eat wood. It's a win-win. But they've been doing this so long that they can no longer survive without each other. The same thing happened when the cells moved inside other cells. At first it was convenient and helpful, but over time it became compulsory. They can no longer live alone. Okay, so there's a skeleton inside each and every cell. As with your skeleton, this supports the cell and allows for movement. These are 3 different types of protein fibers with different functions. They're throughout the cell, and we'll go through the particular functions of each. 3 different types of proteins for different functions. Their jobs are pretty self-explanatory, so I've added no additional information to this slide. This is what those fibers look like. Why would cells that don't move have cilia? You have cilia lining your respiratory tract. And if you're female, you have them in your fallopian tubes. In these cases, the cilia doesn't move the cell, but moves items across the surface of the cell. In your respiratory tract, the cilia is moving mucus. The mucus is capturing pollutants, such as pollen that you've breathed in, and forces it back up your throat until you can swallow it. How often do you swallow? The cilia in your fallopian tubes are there to assist sperm to reach the egg more rapidly. Both cilia and flagella have similar structure since they're both extensions of the cytoskeleton. The flagella in this photo is on a sperm cell, used to move the cell through its environment. The cilia on the left are on cells lining the respiratory tract. The cells don't move. What are they there for again? To move mucus and trapped particles up and out of your lungs. If you're a smoker, you may have a chronic cough. This is because smoking paralyzes the cilia. The mucus is still there, though, and needs to come up and out. Coughing is the only way left for that. Extra in this case means outside of. This matrix is outside the cells and assists in keeping them together, as well as containing signaling molecules such as hormones and being a support system. We looked at cell walls and capsules on prokaryotic cells earlier. Prokaryotes are single-celled organisms. They'll interact mainly with a non-cellular environment. Many eukaryotes are multicellular, but not all. So the cells need to communicate with each other within the multicellular organism. Your cells talk to each other. Plant cells have cell walls, as we saw in the section on chloroplasts. They can be 10 to 100 times thicker than the plasma membrane and are for support as well as protection. Where would you find each type? Where would you need a tissue to be leak-proof? Remember, cells form tissues. Digestive tract, maybe? Bladder? You don't want those leaking inside you, do you? Where would you need a strong junction? Wherever mechanical stress is high, like the skin and the heart. Sure don't want that falling apart. Where would you need channels open? Wherever chemical communication is needed, such as to coordinate a heartbeat. Plant cells. The bottom section here shows the plasma membrane as a yellowish thin line. Note that the cell wall in this picture is showing the walls of 2 adjacent cells. Between those 2 walls is a layer of sticky polysaccharides that glues the cell together. That's dark brown. But the cells are not isolated from each other by those cell walls. Note the pores here. They're called plasmodesmata. These numerous pores form a circulatory and communication system connecting plant cells. See how the cytoplasm and plasma membrane extend through? Through plasmodesmata, water and nutrients and chemical messages can pass from cell to cell. This is just a study aid. Use it or not as you choose. These are cells that line the inside. The dark spots within them are the nuclei.